All right, today what we are going to be looking at is Mendel and inheritance. And you all should have already watched the Mendel video, so you got kind of a brief introduction to who the man was. What I'm going to do is I'm just going to ask you to write down what's in red, and that's going to be the important stuff. So if you feel like I'm going too fast, just please push pause, and then you can pick up when you're all ready to write some more. So let's talk about Mendel. So in genetics, there's been a pretty long history. Way back when, there was Aristotle, and people knew that the man and the woman both contributed to create offspring, but back then, of course, they thought that man had more of the important attributes that they would give to the baby. So the man had the vital essence and the woman just really contributed matter. And so, back in those days, it was really ideal that you would have a male baby. It would resemble the father, and then it wouldn't be as great if you had a girl. But in the 1800s, finally, we started to realize that there were new ideas about how genetics were passed on from parents to offspring. So it was this new idea called blended inheritance. So it said if you take a man and you have a woman and they have a baby, then that baby is going to be a blend of the mom and the dad. So if dad is really tall and mom is really short, then baby's going to be somewhere in the middle. But again, we know that that's not necessarily true. So research showed that that was incorrect. And now we come to present day. And we have had observations and experiments, and now we know that traits are passed on to babies through these discrete units called genes. And it all really came from Mendel, who was working as a monk, and he had all this time on his hand, and he was experimenting with pea plants and was finding out that there were some really amazing things going on as far as what traits were passed on to the next generation. So, from just to recap that video, he was experimenting with pea plants. Sometimes pea plants are really well known as far as they can self-fertilize. So you have an offspring that is going to look exactly the same as a parent. It's a purebred. So you had the round pea plant fertilized with itself, with a round pea plant. So it created a purebred plant. Looks exactly the same. But what Gregor Mendel was doing is he was actually forcing different types of pea plants to cross-pollinate with each other, and these would make hybrids. So the offspring that would arise actually had different traits than the parents, and he was really interested by this. And what he found out is that sometimes a trait would get lost. So in that generation, the babies would not have similar traits to the parents. And he's like, well, what happened to that trait? Why don't the baby pea plants have that look to it? And so then what he did is he took those hybrid babies and he made them self-pollinate, and lo and behold, all of a sudden the trait that was lost reappeared. And so it really got him thinking about, how is this working? How is genetics working? So he concluded that an individual's characteristics are determined by factors that are passed from one parent to the next generation. There are some kind of non-mixable factors that are controlling the traits. and he called it genes. So there's no blending, it's just these discrete units called genes. And each gene is made up of 2 alleles. And so it's just different forms of the same gene. So remember, when we're born, we got half of our information from mom and half of our information from dad. And so we got 1 allele from mom and 1 allele from dad. And so that created the gene that we then show. And what Mendel realized is that some alleles dominate over other alleles. For example, both of my parents have brown hair, but I have red hair. So they have the red hair gene. It's just that their brown head gene dominated over the red hair gene. But then here I am, and I just happen to get both of the red hair genes. So it shows. So what we're going to do here is we're going to briefly talk about what did Mendel figure out and what were his laws. So the first one is he did realize that there was this law of segregation. So alleles separate into different gametes. Remember, you don't want a full set of chromosomes in the sperm or in the egg. You have to separate them. So 2 alleles would separate so that we would only get 1 from mom and 1 from dad. You don't want 2 alleles. You only want 1 from each parent. So then that's going to show correctly. Okay. And then he figured out that there was this principle of dominance, that if you have a gene, you could be a carrier for an allele that just doesn't show. So one of your alleles could be dominant over the others. And if an allele is dominant, then that is going to be expressed. So if you have a recessive trait, then it's only going to show if both of the alleles you got are recessive. For instance, for red hair, I got both recessive alleles from my parents. So they carried it, they didn't show it, but then they gave me both of the recessives. So both together, I express the red hair. And then there was this law of independent assortment. It just means that each gene is inherited separately. So if you look at this picture, you're not going to get the white color with the long tail always. Okay, it's going to be separated. So you can have white cats with a short tail. You can have white cats with a long tail. You can have brown cats with a short tail. You can have brown cats with a long tail. So each of these genes is inherited by itself. They're not inherited together. All right, so here we have our genes. These are a segment of DNA that controls traits by the production of particular proteins. So I have protein A, so my hair is red. If you got the DNA that coded for protein B, you would have brown hair. If there was really not that much protein at all, then you would code for blonde hair. So a gene is just what has all the information that makes our proteins, and then it makes us look the way we do. So remember, we talked about alleles, an alternate form of a gene. You're going to get 1 allele from one parent and 1 allele from the other parent. So for instance, can you roll your tongue? I can. And so I have the big R allele. I can roll my tongue. If you can't roll your tongue, that means you just have the little r's, and that's fine. So some alleles are dominant. They mask another allele, and then some alleles are recessive. So in order for you to show a recessive allele, you should have gotten 2 recessive alleles from each parent, and that's how you're going to show it. So what we're going to do here is when we're talking about genetics, we're going to use a lot of new vocabulary. So the first one is genotype. So the genes for a particular trait that are carried by an organism. we get one allele from mom and one allele from dad, and that creates our unique genotype. So sometimes you can have two different alleles, and that's called heterozygous. For instance, we were talking about tongue rolling. So if you got a big R from one parent and a little r from the other parent, then your genotype is heterozygous. So when we're talking about genotypes, we're just going to be talking about what letters code for your gene. If you got two of the same alleles, it could be you got a big R from one parent and a big R from the other parent, you're homozygous. They're the same. They're big R and big R. If you got a little r from one parent and a little r from a different parent, still homozygous. They're still the same. So genotype is just the code, basically, the letters that talk about a particular gene. Now, when we're looking at our genotypes, we are going to kind of talk about what does that genotype mean. If I have big R, little r, what am I going to look like? What is that going to express? And that's called the phenotype. The expression or outcome of a gene based on the alleles that are carried on the chromosomes. So, did I get the genotype that codes for blonde hair? Did I get the genotype that codes for brunette or for red hair? It's just the words that we're going to use to describe the letters. So, genotype, letters, phenotype are the words that describe it. All right, so let's just look at some of these examples here. Which of the following are dominant? Now, when we talk about dominant, we are going to use big letters. So the D is a big letter, the L, the N, the R, the S, they're all big letters. So that means we're talking about a dominant allele. What about if we're talking about a recessive allele? Recessive just means we're using little letters. So we have the N is little, the D is little, the R and the K, they're all little. So they are all recessive. All right, now remember when we talk about homozygous and dominance, so homozygous means the same, dominant means big letters. So which one of these are both the same and they're both big? We can see that we have big A, big K, and big T. So they are all the same and they're all big letters, so that's called homozygous dominant. What if we're talking about homozygous recessive? Again, homozygous means the same and recessive means little letters. So we have little e, little q, and little w. So homozygous recessive. They are the same letters, but they are little letters. And then if they're ever talking about, what is somebody's genotype if it is heterozygous? Heterozygous just means they're different. So you have big D, big A, big G, little g. Those ones are all heterozygous because you have a big letter and a little letter. So they're different. So what we're going to do here is we're going to use our genotypes to create what's called a Punnett Square. So say two people want to predict the probability that their kids will get some trait or, maybe even a genetic disease. What's the probability with that? So what we're going to do here is you're going to need mom's genotype and you're going to need dad's genotype. And putting those two together, you're going to make a Punnett Square. You're going to predict if this is mom's and this is dad's, what are the possible outcomes for our babies? Well, if we look right here, we can see that the big A is dominant over the little a. So all of the offspring are going to be red. There's no chance that they're going to have blue babies. Usually mom is down the side and dad is on the top, but that's not too big of a deal. So the outside letters are mom and dad, and the inside letters are what are the possible babies? What are the possible genotypes and phenotypes that our babies will have and possibly look like? So in this example, we can see that the possible genotypes, just looking in the middle, we have 100% chance that our babies will be big A, little a. That's the only possible genotype that they can even have. Phenotype is just, what does big A, little a mean? It has a big A. That dominant allele is going to mask the recessive allele, which means that 100% chance our babies are going to be red. All right, and that's where we get to doing the fun stuff. So doing these Punnett squares, we're just going to focus on what is called a monohybrid cross. We're just going to explore a single gene outcome. So what we're going to do here is we're going to look at hair color. And we have, if you get a little b, you're going to be a blonde. And if you have a big B, you're going to be a brunette. So what we're going to do here is we're going to cross big B, and little b, little b. So you put, you're going to put mom on the side, and you're going to put dad on the top. So here I'm just showing you have the possible sperm that is going to bring an allele to the baby, and then a possible egg that is going to bring an allele to the baby. So when you do your monohybrid crosses or your Punnett squares and you fill it in, you always put the big letter first because that's a dominant letter and it's going to mask the little letter. So when we say, well, what are the genotypes? Do we have possible little big B's in that cross? No, there's no chance. Do we have big B, little b's? Yep. We have 4 out of 4. We have 100% chance that our babies will get the big B, little b genotype. And are there any little b's? Nope, no chance of that. So what does big B, little b mean? It has that big B. So that big B masks the little b. And remember, the big letters are dominant. So what does a big B mean? Well, it just means that all of our babies are going to be brunette. There's no chance of the blonde genotype showing in our baby. So we have 100% chance of brunette-haired babies. Okay, so let's do a couple of examples here before we're finished. So here we're talking about a dominant trait is a big H, meaning that our baby could get a high metabolism, or there is the recessive trait, a little h, which just means that our baby could have a normal metabolism. So nothing too crazy here. When you're doing your squares, remember you always have to do your Punnett first before you answer any questions. And the big letter always goes first. Obviously in this box you have big H, big H. If you put the two in the next box together, you have a big H and a little h. Go down to the next box and you'll see that you have a big H and a big H. And then that final box has a big H and a little h. So what are our possible genotypes here? If we look at this square, we can see that we have a 2 out of 4 chance, or a 50% chance, that our baby will have a big H, big H. And then we also have the same. We have a 50% chance that our baby will have a big H, little h. So what does this mean? What does the big H mean? Because remember, even though there's that little h over there, it's with a big H, and he's always going to mask the little h. So all of our genotypes have a big H. What does that mean? Well, if we look up there, we can see that a big H means that we have a 100% chance that our babies will have a high metabolism. So genotypes are the letters, And the phenotypes are just, what do those letters mean? All right, one more, and I promise we're done. So here we're going to look at pea plants. The dominant trait is a big T, meaning that they'll be tall, or there is a recessive trait, little t, where they will be short. So here what we have are we have two pea plants that are heterozygous. So down the side we have big T, little t, there's mom. And across the top we have big T, little t, that's dad. And so then in the middle, if we put them together, we can see that we get big T, big T, big T, little t, big T, little t, and then a little t, little t. So what are our possible genotypes? We only have 1 out of 4 that is big T, so a 25% chance. We have a 50% chance that our babies will get a big T, little t. And then there we have a 1 out of 4 chance that our babies will get a little t, little t. There is the recessives. That's the only time that you're going to see the recessive trait show, is if they both have the little letters. So what does this mean? What do these letters mean? If we'll notice here, 3 boxes have a big T. And remember, those big T's always mask the little t's. So we have a 75% chance that our babies will be tall. But we can see there that we do have 2 recessives together. So that is, that should be 1 out of 4, should be little t. So we have a 25% chance that those will be short. So what I'd like you to do is work on the worksheet that I've attached. You really need to use your notes. I hope you've been writing down your notes and paying attention, and we're going to be working on Punnett Square stuff for a little bit. So if you're really struggling, shoot me an email, please, and then that will help guide me for what we need to do next. So let me know if the worksheet's working for you. I hope that you can just click in boxes and fill in the genotypes and do the best you can, guys. That's all we can do. All right. Miss you guys lots. I hope you're doing well, and we'll talk to you soon.